Good morning, everyone, and welcome to the second session, AI and Computational Biology. My name is Nasun. I'm from Whitehead Institute, and I'm very glad to be here today with everyone to enjoy this wonderful symposium. And in this session, we have 4 outstanding speakers working at the intersection of computation, AI, genomics, and medicine. So some notes here, and our keynote talk will about 35 minutes with 5 minutes questions. And also each of the following talks will run 25 minutes with 5 minutes for questions. So after each talk, we'll have a brief recognition ceremony. And okay, let's get started. So our keynote speaker today is Dr. Manolis Kellis, professor of computer science at MIT, a member of Broad Institute, and head of MIT Computational Biology Group. And on a personal note, I'm especially glad to introduce him because Manolis was my PhD advisor at MIT. And he has led some of the large-scale genomics efforts of the past 2 decades, including ENCODE, Roadmap Epigenomics, and the GTEx project, with work spanning regulatory and comparative genomics applied to Alzheimer's disease, cancer, and many other diseases. He's a recipient of the Presidential Early Career Award and NSF Career Award, among many other honors. So today he will speak on AI for genomic medicine. Please join me in welcoming Dr. Kellis.
It's a huge pleasure to be here. I realize just how close the Chinese culture and the Greek culture are, besides being fantastic, amazing civilizations that have really shaped the world as we know it. I'm also usually the last person to enter a conference room. And it was amazing to find 20 other people who were waiting to enter after me. So it's, I just hadn't realized just how many similarities we have. So it's a huge pleasure to be here. I spent several summers in Shanghai, teaching students there. And I've also visited Beijing and Xi'an and all of these extraordinary parts of China. So it's really a huge pleasure to be here. Most of my postdocs are Chinese, as you can imagine. I didn't know if I was in the right room because just seeing only Chinese people did not mean anything about this being the Chinese Life Sciences Symposium, just because Harvard is so extraordinarily well diverse and populated with talent from all over the world, including so many first generation, second generation, also immigrants from China. I'm an immigrant myself. I grew up in Greece. I came here as a foreigner. I learned to speak English. I learned how to do science. I learned how to, I would say I learned how to think here at MIT as a student. So it was a huge honor to, of course, receive the prize from President Barack Obama, another sort of, son of immigrants here. And I just really hope that the US will come to its senses and recognize the extraordinary importance of immigrants in really shaping this country. So anyway, truly a huge pleasure to be here. I think the contribution of Chinese scientists to science has been inordinate and extraordinary. And it's a pleasure to be here. So what I'll talk about is AI for genomic medicine. Basically, how do we use the power of AI and all of this extraordinary advance that has happened that's really shaping the world to advance our deep understanding of biology. And I don't want to be provocative. In fact, I do want to be provocative and say that everybody's so focused on LLMs. Oh, LLMs are going to change the world. And I think LLMs are wonderful and they're great, but it's not what's going to solve genomic medicine. It's not what's going to sort of transform our understanding of biology as we know it. They're amazing reasoning engines, but they think like humans think. And in order to truly enable AI for genomic medicine, we have to think in new ways. We have to go where no human has been. In fact, I'm going to jump ahead a little bit. So basically, if our goal is to sort of change the way that we understand precision medicine. If our goal is to change the way that we understand drugs and disease and circuitry, then we need to lift the humans up. We need to go beyond our traditional thinking. And the example that I want to give is AlphaGo and AlphaZero and Deep Blue versus humanity. So Gary Kasparov is, someone that I've interacted with a lot. His kids and my kids are the same age, so we've played chess before, and it didn't go well for me. But he's someone who really represented humanity against AI. And at the time, I would say that it wasn't really about AI. It was like humans deciding how to score chessboards. It was humans decided the minimax algorithm itself. It was very different from what it is today. The AlphaZero of today is very different and much more similar to AlphaGo. What did AlphaGo do? AlphaGo basically said, forget with human play. Let's just play from scratch. All it knew was the rules, and it had 3 days of self-learning. Lee Sedol, representing humanity, had 3,000 of years of human history playing Go, and all of the philosophy of Go and all of the strategies and everything that we thought we knew about the game. But the end result is that humanity was humbled, and Lee Sedol retired, basically saying that it turns out that we humans really did not understand the game of Go after thousands of years. And this is sort of the context in which I want to place this. I don't want LLMs to be just like glorified scripts that basically do the stuff that we tell them, just faster and cheaper, and fire all the humans to come back a year later and realize that everything's broken because the AI was just simply repeating whatever we told it to do. What I want is to elevate humans. I want AI to sort of take us to places that we've never been before. And the secret that we're using in my team and across many labs across the world is the concept of latent embedding representations. It's about understanding the geometry of meaning. And this is where the two different contrasting representations of gravity come in. So Newton basically came about and said that there's this extraordinary unifying force that acts on apples falling on Earth, just like it acts on the heavens with planets orbiting the sun and moons orbiting these planets, where gravity is a force that acts at a distance proportional to the product of the two masses and inversely proportional to the distance, the square of the distance between them. So Einstein came along and said, what if there's no force? What if gravity is just a consequence of the geometry of space, a consequence of the curvature of the space-time continuum? And this was enough to explain the action as a consequence of the geometry. And it wasn't just enough to explain the events that were observed at the time, but to predict more than 100 years later the observation of gravitational waves that we only observed through the LIGO Observatory a few years ago, to predict the exact curvature of light surrounding black holes and eclipses, to predict the exact slowdown of atomic clocks that had not been invented for another few decades after that. So the power of understanding the geometry is that it gives us a meaningful representation. It gives us a meaningful understanding of the underlying processes. So the way that I want to think about the Aleph is this unifying force, this unifying representation that is able to compress the universe, similar to the story by José Luis Borges in 1945, that described this Aleph as this point in space, point in the universe that contains within it all other points in their full glory. And everybody who would see this Aleph, who would see this sort of merging of information, would basically be seeing this vision, overwhelming vision of infinity. So this is where I want you to think of AI as a prism that basically brings through its representation all of the knowledge that we have. So when I ask you to think about the word apple, you're thinking about A-P-P-L-E. You're also thinking about the sound apple. You're thinking about the shape of an apple. You're thinking about perhaps an apple a day keeps the doctor away, about the 100 calories, about the nutritional value, perhaps the seed, the biology, the taste, the smell of a freshly cut apple. So you have this multimodal representation of an apple. natively inside you across all of your senses. If I ask you to think of a gene, let's say apolipoprotein E, you have an intuitive representation of that gene. Maybe its association with Alzheimer's, maybe, the fact that it serves as a lipid transporter and all kinds of other stuff. But that's the representation that I want to elevate. That's where being able to integrate every single data set that has ever been observed about ApoE can basically elevate our intuitive understanding, create this geometry of meaning, create a landscape of every gene linked to every other gene across all of biology, all of chemistry. So the same way that an apple is projected to a bunch of places, maybe the way that we understand genes is through this knowledge graph, basically telling us how every gene is connected to a bunch of different diseases. It is connected to the phenotypes that it is associated with. It's connected to the drugs that are known to target that gene, the anatomical structures where that gene is expressed, the pathways that it's a part of, the cellular components, the molecular function, the biological process, the exposure, maybe the structure of that protein, maybe the vector of expression of that one gene and all of its isoforms across every single cell experiment that's ever been carried out. So that's what I think lies at the core of the AI revolution. It's not the glorified scripting that LLMs allow us to do, which are, again, wonderful. But that's not how we're going to get super intelligence. The super intelligence comes from painstakingly building these meaningful representations, these meaningful foundational models that are able to capture through the designing of loss functions, of architectures, of pretext tasks, of training data sets that basically then allow us to really elevate our own understanding of biology. And I think this ability to not just think about the problem right in front of us, the grain of sand that each of us is carrying through the edifice of science, but to be able to sort of step back and see every scientific paper that's ever been written and sort of how it shapes every one of these genes, every experiment, every chemical, every patent, and so on and so forth. So that's where modern AI is able to truly make a difference. And the key to all that is the geometry. This is what we call representation learning. It is the ability to not only think about the pixels of an image, the X of the data, to not only think about the Y of the labels, whether there's a dog, a cat, a bird, or a bird in that image. Because predicting from the pixels alone through a fully connected network the objects of an image is, yes, solvable because these functions can approximate anything. But the problem is that the size of that network is bigger than our known universe to be able to actually understand what we do intuitively. So how do we do it, and how can AI do it? The way that we do it is that we don't think about pixels. We abstract the pixels through a series of abstraction layers, a series of representations. So we combine together multiple pixels into lines. We combine multiple lines into shapes, multiple shapes into objects. And it is through that process of abstraction that we can now compute here in the latent space, in the meaningful vector representation. And if we do it right, that vector representation can answer, just like the Aleph, just like a prism, just like our own understanding of an apple, it can answer almost any question about a gene, a protein, a cell, a region of the genome, a regulatory motif, a genetic variant, and so on and so forth. So what I'll do today is that I'll show you a series of examples spanning all of these different data types for how representation learning is changing the way that we think. The same approach that applies to images of basically being able to learn this latent representation applies to chemistry. Every atom in isolation has some small amount of information, but it is in the context of the other atoms that they take meaning. A pixel in isolation could mean either the shirt or blue sky, or we don't know. It is through the context of all of these other pixels nearby that it takes its meaning. In the same way, a carbon atom alone is very different from a carbon atom in the context of all of the neighboring elements. So that basically means that I can now, the same way that for images, I could basically go through and learn recurrent patterns, I can basically learn recurrent subgraphs at the level of a chemical that are then associated with the function of that chemical, because that's the only way to learn repeating patterns. The same way that for language, we start with tokens, and these tokens have repeatable meanings that change in the context of other tokens, but then become meaningful in reproducible ways. The same way we can now understand chemistry from the atoms up. We can understand protein structure, where the building blocks are now amino acids. Again, every amino acid has certain properties which are informative, but they take meaning in the context of other amino acids. Similarly, nucleotides individually have very little meaning, but in the context of other nucleotides, they take meaning. And the same approach applies both to human language and to DNA language. And we can do this also at the level of scientific papers. We can basically take every scientific paper that's been written about apolipoprotein E, ApoE, and basically create vector representations in this 1,500 dimensional space of all of human language for every one of those papers. And we can understand the clusters untangling the different functions of the ApoE protein. And the same thing applies to cells. The same thing applies to transcriptional data or epigenomic data. We can basically take the vector representation of 20,000 protein coding genes and basically represent every cell based on that vector. And there are many ways to do that, but one way is to basically compress that vector into a meaningful set of perhaps linear dimensions through singular value decomposition or principal component analysis. And then we can now start reasoning about the dimensions of that vector. We're no longer reasoning about a single gene, we're reasoning about meta genes. We're no longer reasoning about a single cell, we're reasoning about meta cells. And using that, we can now start recognizing the structure of that latent landscape. So here I'm basically showing you our own data across 2.3 million cells from 400 individuals in 6 different brain regions, where you can basically now look at the latent embedding of every one of these cells and the position of these cells relative to each other in this, of course, two-dimensional representation projection of this high-dimensional space so that we humans can access it and understand it intuitively, basically reveals clusters that turn out to be extraordinarily biologically meaningful. And we can go a step further. We don't have to sort of understand things unimodally. We can understand things multimodally. We can basically co-embed chemical structure and protein structure and the patents that were written about the drugs that target those proteins and the domains of these proteins. And we can do that for patient records. We can basically co-embed the vector representation of the doctor notes, the language representation, along with the quantitative variables, the lab tests, the imaging data, and so on and so forth. So we can combine these representations to, again, create this aleph, this prism, this combination of all of these abstractions. So let me walk you through a number of examples, first in single cell space, then in protein structure and function, and then in chemical space, which are the different components in order to be able to enable this AI for precision medicine that I promised. So here's what these representations look like. What these representations are effectively vectors in this very high dimensional space. Every one of these dimensions can be interpreted in some cases. For example, in the case of gene function, one dimension might represent how associated with lipids it is. Another dimension might represent how associated it is with the endoplasmic reticulum. And another dimension might represent how expressed it is in liver-like cell types, and so on and so forth. So basically, these representations are such that they try to capture the true dimensionality, the true lower dimensional manifold where the data truly lives. But they, in some cases, can be interpreted so that we humans have an intuition about what that actually means. And that basically means that we can now start reasoning geometrically about the gradients in that latent landscape. We can start reasoning about the archetypes. We can basically start thinking about summarizing of scientific papers as vector algebra, rather than having to go back into the language. We can think about what it means for multiple components of a chemical to be combined together, because the spaces that we construct have compositionality, and therefore addition in that space has a very meaningful interpretation as a combination of the function of those molecules. And that's where cognitive cartography comes in. That's where the ability to actually see these latent landscapes. So let me walk you through an example. So we did exactly what I was describing in the previous slide. We basically created a vector representation for the function of every gene that incorporates within it the entire graph neural network of this knowledge graph connecting every gene to every one of those biological constructs. And that allows us to now learn a latent representation, not just for every gene/protein, but also for every disease, every phenotype, every anatomy, every biological process and exposure. And this is what the space of protein structure and function gives us. So, because we have co-embedded both the knowledge graph representation of these proteins, as well as the structural representation of these proteins. And what that gives us is now a cartography of all of human biology, where we can basically take every gene, and instead of like looking at an index or looking at a database, we can basically look at a map of how these genes are related to each other. We can also do the same thing for the drugs that target these proteins. We can basically construct a landscape that contains, in the same way, chemistry, every single drug that has ever been defined. But the most interesting part is when we combine the two spaces together, where we basically take the geometry of all of chemistry, and we can take the geometry of all of protein function, and we can use links, anchors, based on the known pairing of a protein and the drugs that target it, to reshape, to learn a translation function between, let's say, the 1,000 dimensional representation of a protein function and the 700 dimensional representation of a gene function. So we can basically train a neural network that translates between the two into a common representation. And when we do that, really magic comes out. So what we can see is that we can now see the entire landscape of drugs, every single drug that has ever been synthesized. We can now start seeing how they cluster in that latent landscape. And what we see is that when you compare that to experimental information about several of these drugs that have actually been tested, what you can basically see is that the result of those tests matches what the continuum of the latent landscape would predict. So we can now use that to basically interact with these directly, to sort of navigate these latent landscapes, to be able to understand groups of these functions. And we can do the same thing with patients. Like in the patient landscape, you can basically see a continuum between multiple axes of variation, where similar patients are grouped based on their conditions next to others. And we can do the same thing in cells. We can basically find that there are biologically meaningful relationships being captured here. So to dive through this example, we took, again, 2.4 million cells across 400 individuals with and without Alzheimer's disease. And we basically said, can we understand how these phenotypes map at single cell resolution? Because the problem that we have is that everyone in the field of single cell analysis makes the implicit and unstated assumption that we have cases and controls, and we're comparing two conditions, most of the time binary. And implicitly, again, we're making the assumption that every single cell from an Alzheimer's person is 100% Alzheimer's. And every single cell from a control person is 100% control. And that every Alzheimer's person is 100% Alzheimer's. which is truly not the case. We are continuums. Every one of us is a continuum. And the phenotype itself progresses over time. Alzheimer's take 10 plus years to truly manifest from the beginning of the dysregulation. So instead of making that assumption, we basically say, okay, let's start with a binary phenotypes. And now let's go into neighborhoods of cells. So we're leveraging the geometry of that latent landscape. And what we're basically doing is coloring every neighborhood in that geometry according to the number of donors whose cells were Alzheimer's or control. So if in this neighborhood, the vast majority of cells came from Alzheimer's individuals, that will be a red neighborhood, an Alzheimer's case-like neighborhood. If that neighborhood is instead very blue, it means that the vast majority of cells in that neighborhood came from control individuals. We can now start ignoring, after this step, we ignore the original binary phenotypes. And we now simply have continuous phenotypes for every neighborhood. And that basically means the cell in that neighborhood will now be annotated, not by whether it came from Sally and she just happened to be diagnosed with Alzheimer's, or from Susan who happened to not be diagnosed with Alzheimer's, but whether the cells came from 400 donors that were primarily of one phenotype or another phenotype. And therefore, we can now start coloring every neighborhood according to the annotations of the donors. Many donors, not one donor. And therefore, even if there's a mistake in the annotation, or if the heterogeneity of Alzheimer's fooled us into thinking it was Alzheimer's instead of frontotemporal dementia, or some other neurodegenerative variation, we can now say, well, there are some neighborhoods that are reliably Alzheimer's-like and some neighborhoods that are reliably control-like. And now we can go back and reannotate every cell according to the neighborhood that it's a part of. And the most exciting part is that we can now go back to the individuals. And instead of saying that this person is 100% Alzheimer's, we can say, well, their cells appear to be distributed a little further to the right of that distribution. And therefore, I'm going to give them a score that they're like 98% Alzheimer's. And this one here, even though they were annotated as blue, turns out that many of their cells were red. Maybe they're 75% Alzheimer's for that cell type, for microglia in this particular case. And this red individual here, well, all of their cells were in this very blue region. So that individual will be annotated as very control-like. So this changes things completely, because yes, we do see that the distributions are shifted, but they're highly overlapping. And we can do this, as I mentioned, for the Alzheimer's phenotype in microglia. And this is what I showed you on the top right. But we can also do that for Alzheimer's in every single one of the cell types. And therefore, this individual, let's say Sally again, has her microglia in very red neighborhoods, but her oligodendrocytes are sitting in very blue neighborhoods. And her excitatory neurons are in fact very blue. So that allows us to now start taking this monolithic view of Alzheimer's as A, one disorder, B, a single axis of variation, C, only binary yes, no, and D, every cell is exactly identical. Two, there's a continuum of manifestation of the disease across different tissues, across different cell types, across different biological pathways, across different cell types, and also across different phenotypes. I don't have to just do this projection for Alzheimer's disease. I can do it for tangles and amyloid beta and neuroinflammation and cognitive impairment and the speed of cognitive decline. And every one of them paints these neighborhoods in a different way. And every one of them does manifest in these single cell neighborhoods of the brain, which is, again, mind-boggling. So what that basically means is that I can now start looking at progression in this latent landscape of cells. And I can use that progression to guide interventions to basically say, what will it take to nudge the cells in this red neighborhood to become more blue neighborhood-like? And to give you an example of that, we can basically go and study these frontiers between red and blue. And what we're finding is, again, very unexpected. I did not expect to see this. What we find is that if you cluster all of these neighborhoods, this is just looking at one cell type, microglia. if you cluster all of these neighborhoods, what you see is that there's a very long border between health and disease. It's not like, oh, all of the blue is on one side and all of the red is on the other side. No, for every one of these regions, there's a blue-red frontier. And in fact, if you look at the clustering, A3 and N9, these Alzheimer's-like, Alzheimer's-enriched neighborhood 3, and these control-like, normal-like neighborhood 9, are in fact the two closest neighborhoods to each other, except for one is Alzheimer's, one is control. That's a huge opportunity. We can now look within this neighborhood. What are the differences? And what we're finding is that there's distinct hallmarks of Alzheimer's, distinct genes that are differentially expressed in every one of those neighborhoods. So the hallmarks of Alzheimer's are falling out right in front of our eyes. We can now start interpreting all of these frontiers as distinct axes of variation. And that basically means that we can now start designing therapeutics at the hallmark level, not at the monolithic level. And when it comes to understanding which patients to prescribe these to, it gives us also a way to get at personalized medicine. You can basically say, what is the neighborhoods that we need to sort of shift in every one of those patients? In the case of cancer, this is very exciting because cancer has accessible cells. This is an example from cancer where, again, we use this cell-projected phenotypes approach to basically annotate all of the cells in the single cell landscape according to this, in this particular case, lung adenocarcinoma score. And then that gives us scores which are validating in different cohorts and which are clustering spatially across different neighborhoods. So you can basically now see that these landscape-based neighborhoods in this latent manifold of gene expression are in fact localizing to specific regions of the spatial transcriptomics profiles. And if you look here, what you basically see is that there are some regions in the H&E staining imaging, which when overlaid with spatial transcriptomics, have the single cell signatures of tumor in yellow and control in blue. And you can also see this border here, which is very mixed between blue and yellow, and this infiltration that appears to be happening. We can find specific cell-cell communication patterns that are happening exactly at that frontier. So this is very exciting. But do we need to get the brain of a person to say what drug we want to give them? People like to hold on to their brains until they're done using them. So what are we going to do instead? Well, it turns out that biomarkers like urea, AMP, benzoate, localized to very distinct regions of that latent landscape neighborhood. So we can basically use the cellular atlas as a translation coordinate frame between the phenotypes of the patients, between the molecular level functions of the genes that are dysregulated in every one of these regions by basically taking the vector representation of function for every gene, aggregating it at the cell level based on the genes expressed in that cell, and aggregating it at the neighborhood level based on the cells within that neighborhood, quantitatively based on each of those. And that gives us a functional representation for every single cell and every single neighborhood. So when I see differences between Alzheimer's and control, I don't even need to do Go analysis anymore. I can look directly at the embedding representation of function and look at the gradient for what are the dimensions of function that are changing along each one of those lines. And I can map using the biomarkers of each individual, which might be imaging, they might be genetics, by aggregating along each of those hallmarks and pathways to basically understand how the function of those individuals is changing. So how do we deliver on the therapeutics now? What we need to know is what are the drugs that are going to be nudging the cellular state from this functional state to this other functional state, from disease back to healthy. And to do this, we've basically trained these multimodal foundation models for joint representation of protein structure, chemical structure, and language. In this particular case, this is Procyon, work that we did jointly with Marika Zitnik, who's here at Harvard Medical School, to basically look at the ability to speak the language of protein structure. And then we can go and sort of talk to the proteins and say, okay, I'm giving you a protein sequence or a protein structure. And I'm speaking to the AI that now has this multimodal understanding. We can do all kinds of wonderful things. And we can also do the same thing with chemical structure to function. So we've basically taken drug molecular fingerprints, this 2000 dimensional vector, protein representations that capture some aspect of structure, some aspect of function through Pro-T5 in a 1000 dimensional vector. We've taken protein domains in this 800 dimensional vector, and we've taken also patents that describe the function of every one of those drugs. Why? Because they really guide the regularization. Multimodal models are much more robust to these mistakes and this overfitting. And we're projecting them jointly using this joint representation learning, this joint latent space, under the additional constraint that I can re-represent and reconstruct the drugs from the embedding vector directly. And therefore, we learn an interpretable latent space that we've basically now applied to 70 million drug target pairs. So it was an enormous engineering challenge to construct this. But the end result of that is that we now have the world's best representation of chemical function and structure. Now, toxicity is just an emergent property of learning this landscape. We never told it about toxicity, but what you can see is that toxicity clusters in very minute ways within this latent space. We're able to also just identify non-binders from millions of molecules. And we also are able to build this robustness score that basically tells us if I move along the latent landscape, how much is the prediction moving? And the smoothness tells us so much about the confidence of that space. And we're now able to predict at nanomolar resolution the binding affinity of chemicals to proteins across this entire matrix of that space. And we've basically validated this nanomolar resolution for many of the novel compounds. And we're able to now create these, of course, agentic wrappers, these glorified scripting, to basically discover drugs systematically by leveraging this latent landscape. We say find a combination of approved post-phase 1 clinical trial drugs that can treat Alzheimer's. And of course, our agents are able to go and sift through all of the literature, all of these data sets, run a bunch of programs, etc. But the true intelligence comes from the quality of the latent landscape. And of course, we're testing now those systematically. We have a whole drug discovery pipeline, collaboration with Brad Pettelute at MIT. And we're basically testing these organoids led by Martin in my lab, Martin Walwen, where we're basically taking hundreds of donors in multi-well format, testing combinations of drugs and observing the results of changes in the firing patterns of those neurons using multi-electrode arrays, using also single-cell profiling to see are the shifts that we see in the latent landscape in the organoids matching the ones that we saw in the postmortem patients. And the last part is that we really want to let the humans in. We want to build these maps to engage our geometric reasoning. Humans are amazing at understanding maps, understanding cartography. Here I'm showing you 3 million articles from the New York Times where you can basically directly see that latent landscape. You can directly interact with it, something that you would never be able to do using just a chatbot. We're able to do this across every scientific paper written by MIT authors, every grant, every class, every startup, every loan across hundreds of thousands of loans. Look at knowledge evolution across thousands of papers. We can look at our own lab and how we're progressing over time, how our students are thinking, how they're meeting, how they're writing, and so on and so forth. And we can also create these multimodal representations, where here I'm basically showing you how the papers relate to the grants and to the physical space from the same. So basically we've created this interactive platform for letting the humans back in, for seeing these clusters, interacting with the lasso tool, speaking with the agents, all in the context of this geometric reasoning. And this is completely natively multimodal, understanding protein structure in a completely native way, understanding images, understanding chemical structure, understanding every modality. And including, these are the meeting notes. We basically have written scripts that now analyze the way that we humans think and are able to now take our ideas from every meeting that we've ever had and then see how these relate to each other. Every conversation of our research team is now automatically augmented with this rich information. We can see for every student, where are they in this latent landscape. For every new idea, who should be working on that? This is automatically dispatched. So again, I think that this approach of thinking geometrically can change the way that AI think. And we can also change the way that we function, the way that we work. This is, for example, our own code base, and basically being able to sort of navigate this latent organization of everything. This is the AI article, the Wikipedia article for artificial intelligence, and you can browse it semantically instead of browsing it in the traditional way. So that's the vision for AI as a discovery partner, not as a glorified script, but instead as learning these deep representations through these latent embeddings across all of these different data modalities. And also AI as a Google Maps that lets the human back in, that gives us this dashboard with which we can navigate our own data. That's where I'll stop, and I'll be super happy to take any questions. Thank you.
Thanks for this insightful talk. I believe everyone can see the power of the AI in the biomedical research. So now, anyone have questions? Yeah, we may have 2 or 3 questions.
So this is the most recent photo of the lab yesterday. I couldn't help. Anyway.
First of all, thank you for the talk, and amazing. To clarify, I'm a clinician. I'm not like a computational science. So probably my question could be just, too naive and sometimes stupid. So let's say you have your AI model for precision medicine running. Now you discover like something is wrong. What's the turnaround time to have that fixed? I mean, and for example, this happens oftentimes. Take Alzheimer's, for example. Like 10 years ago, we all think about A-beta monomer. Now, it shifts over to A-beta polymers. So let's say your AI model was built based on the monomer. Yeah. Now you want to fix that with the polymer, and then I'm sure there's something about the weight to change the weight, change the parameters, whatsoever. What's the turnaround time for fixing?
Fantastic question, extraordinary question. So the question is, when an LLM learns a latent representation for human language, it's very hard to unlearn. So basically, with current monolithic giant billion parameter models, it's very difficult to say, ooh, that piece of information is false. You have to sort of remove it from your understanding. And I completely agree that this is a huge problem for LLMs today, because you don't know where that knowledge is encoded. It is distributed through the multi-headed attention, through this representation that is like spread out throughout the model. The difference in what we're doing is that it's based on primary data. Therefore, when the scientific literature is incomplete, you're able to recognize what are the primary papers that led to this difference in embedding. And in the case of chemistry, for example, the anchoring between the protein structure and the chemical structure is based on known interactors. We need to close the loop. We need to be able to say, okay, great. We've now studied this little part of the chemical space. Let's go in and synthesize many, many more drugs in order to enrich the density of representations in that space. So basically what I would say is that in this geometric approach that we've taken, where the contrastive learning is what has led to the shape of these representations, it's actually fairly straightforward to reweigh based on an additional data set. So the way that we're thinking about intelligence is that it starts with these overall foundation models in each of the domains separately, and then you're doing the contrastive learning to fine-tune them based on the local knowledge. That step is very rapid to alter. And therefore, you have a bunch of new papers that are revealing a different facet of Alzheimer's disease. This will take you on the order of days to retrain, not months.
Do you expect this kind of fixing to be automatic or like a man-in-loop correction here?
I tend to think that similar to the arc of justice, we tend towards truth, that we're not going to be completely shifting away, that yes, we're not going there straight. we're kind of asymptoting towards truth. But in the same way, I don't think that we're going to be dramatically wrong, especially because in many of those data sets that I showed you, these are primary data sets. These are based on experiments. These experiments are not false. The only part that I would worry about is the scientific papers. But then again, we are treating them as point in a vector landscape, and therefore we can reweigh them based on, a paper was shown to be false or, not reliable, et cetera. So basically, because that representation is very explicit, it's not just, feed everything to an LLM and then it'll figure it out. I think it's much easier to intervene. Let's take a couple more questions because I don't want it to be just about one topic.
Yeah.
There's
I think we need to wrap up here, and I think Manolis.
one quick question from a student or no? No student questions?
Question.
Okay, great.
Okay.
Thank you for a great talk. So actually, I want to ask for your comments on scaling law for biomedical sciences, because recently there's a Nature Methods paper discussing what happens if you increase the training data size for deep learning methods like SCGPT or SCVI framework. And their conclusion is we're not getting much by scale up.
Yeah.
So I really want to know your thoughts on this.
It's a fantastic question. The question is about sort of the scaling of data, the scaling of the model size, and then how much better does it lead to. So a lot of the models that we build are fairly shallow. They're not sort of these billion parameter models. They're much more compact. So basically our affinity net model, for example, for chemistry and protein structure is dramatically smaller than many of these giant models out there. And I think that the world is obsessed with the model size. And what I would say is this is like taking a tractor and then scaling the engine without sort of thinking about the wheels and the chassis and sort of where it's going to be running. I think the way that you use your intelligence is so much more important than the sheer size of the model. And there's an enormous cost with running these models that are very often oversized. There's cost associated with hallucination. There's cost associated with, being able to sort of make up things that are just not in the data set that are inferred because you've used a single monolithic model. So I think a lot of the field is moving towards smaller models that are more experts in specific different domains and classes. And I think that dramatically changes your training cost, your runtime cost, your memory footprint, and frankly, the ability to contain the model to be much more grounded and less hallucinating. So again, for single cell data, a lot of the linear models are, fine representations of it. A lot of what I showed you was based on these linear latent embeddings. They're not based on these sort of dramatically multimodal and sort of, language-based models. So a lot of the successes that we found are not simply, let's relegate to the AI, let it figure it out. It's really about human engineers deciding exactly sort of how the structure of the space will be. So I have not been impressed with AI intelligence. I think it's really like, when it comes to architecting solutions, the humans are still leading by far. And we've tried. It's not for lack of trying. They're amazing at language. They're amazing at reasoning. They're amazing at so many little things, but they're not yet at the level of design the architecture, come up with the next representation, et cetera. I think that we still to this day have not been, sort of even matched by these models. So hope for humanity.
Thank you very much.
Thank you.
On behalf of Harvard Medical School, CSSA, I'm very pleased to present this operation certificate to Dr. Kellis.
Gosh, okay, thank you. Let's do the photo somewhere, please. We love photos in our lab. Awesome, thank you. Sorry. Which I would take next.
Okay, let's move on to our next speaker. Our next speaker is Dr. Soumya Raychaudhuri, professor of medicine and biomedical informatics at Harvard Medical School and the director of the Center for Data Science at Brigham and Women's Hospital. He's also an institute member of the Broad Institute. His lab studies the genetic basis of immune-mediated disease using human genetics, functional genomics, and systems biology, with a particular focus on how genetic variation contribute to the autoimmune disease and inflammatory conditions. So please welcome Dr. Raychaudhuri.
Thank you. Yep. Can start playing. What do you guys?
Going to the Zoom.
Sorry?
Zoom.
Enter. Where is it? We're sharing the desktop.
Oh, okay. I got it. Plate. No.
Oh,
Yeah, plate.
Plate?
Yeah.
in
In Windows.
in Windows.
Yeah.
There you go.
And
And now maybe hit play.
Plate. Yeah.
play here. Yeah,
Cool.
Perfect.
and
Perfect.
we can share the
Presenter.
presenter. No, not display. You should share the share the
Just want to share that.
yeah,
There you go.
that's
Okay.
it's fine.
All right. Thanks so much for waiting. Yeah, with all the advances in AI, slide presentations continue to be a challenge. So I'm going to talk to you today about some work that's been going on in the lab, where we've been trying to identify the causal allele in a particular locus, a CD40
autoimmune locus, and how that's really helped us try to understand what the causal cell state for B cells and autoimmunity. So I'm going to just start by talking a little bit about autoimmune diseases, which is the space in which I work. And then I'm going to talk about a protocol that we've developed to try to define causal alleles functionally through genome editing. And then finally, I'm going to talk about how we've applied that to this particular CD40 locus. Autoimmune diseases will be familiar to many of you. These are a set of diseases where the immune system attacks a particular organ or set of organs. So for example, with systemic lupus erythematosus, the immune system attacks the kidney and some other organs such as the skin. For thyroiditis, your immune system attacks your thyroid gland, and so on and so on. And one of the great advances in the last, say, 2 years in the field of autoimmunity is the recognition that CAR T cells can be incredibly effective in a subset of patients with autoimmune diseases. So here are a set of individuals with systemic lupus erythematosus. They had intractable diseases, had failed multiple therapies, and Georg Schett and his colleagues applied CAR T cells to ablate the full population of B cells in these individuals. And you can see that they ended up with drug-free remission. So not only does their disease levels go to zero, they're actually not on any drugs at all. And the technology here is these CAR Ts, which are CD19 CAR Ts, which are wiping out the full B cell population. So an incredible advance in autoimmunity. When you think about it, it sort of means, if this holds for other diseases and for other patients, it means that there's a subset of individuals that we can actually get them to a cure. So this, in our lab, we think a lot about human genetics. And one of the things we've been wondering is, can we define a specific B cell state to target? Because the idea of targeting all B cells in all patients with autoimmune disease is a kind of very aggressive idea. But you could imagine with individuals who have like early stage autoimmunity, maybe if we could wipe out a narrow population of B cells or really understand what B cell state is really driving the early stages of disease, maybe we could come to a more effective therapy. So CD19 ablation is expensive, right? And it's also dangerous. But can we define a more precise cell state? Is there some place along this continuum of B cell development that we can narrowly go after? And maybe genetics can help us understand the role of B cells in autoimmunity and help us define what a key cell state is. So the genetics of rheumatoid arthritis and the genetics of many autoimmune diseases have followed a similar path. They've been very successful. And over the last 20 years, we now have something like 150 risk alleles identified for rheumatoid arthritis, 2 papers in press in Nature Genetics right now. And one of these loci that if you're interested in B cells, you might want to pay attention to is this CD40 locus. So this was a paper actually I wrote as a postdoc, and I've been spending about 20 years trying to figure it out. But I think we finally got there. So this was an allele we reported, and we knew from the very beginning that the SNP was right near the CD40 gene, and the CD40 gene, of course, is really important mechanistically for B cell biology. Those of you on the immunology side will know that when T cells and B cells engage with each other, when T helper cells engage with the naive B cell, activate them, the T cell expresses CD40 ligand, engaging with CD40, leading to B cell proliferation, activation, and eventually memory formation and plasma cell formation. So really important protein in the B cell life cycle in this sort of critical moment of its life. But this locus, like many other loci, have the challenge that many GWAS loci have had, which is that when you look at the SNPs associated with the disease, it's very simple to look and say, okay, there's a gene nearby, and I think this is the attractive gene. But the reality is, most of the time, we don't know the causal allele. You have a whole bunch of SNPs that are in linkage with each other. They're in perfect LD with each other. And understanding which one is actually driving the phenotype versus which one is a sort of a passenger is really unclear. So we don't often know the causal allele. In practice, we don't often know the cis gene, though we think we have a good guess. And then very importantly, we don't often know what the transgenes are. We might know the SNP acts on this gene right nearby, but we don't have that module of genes downstream that we have a sense of. And you really need to have a sense of that module of downstream effects to really understand what the effect of the SNP is on cell state, which is mechanistically what's leading to disease. So I'll talk a little bit about our platform that we've been using to sort of take this problem apart. And so one idea that we've been chasing and many others have been chasing is the idea that you can do SNP editing to define what the causal allele is. So if you have a whole bunch of SNPs in LD, maybe you can edit each of those SNPs and figure out which one is the one that leads to the molecular phenotype of interest. Now with base editors, prime editors, you can really do these single SNP resolution edits, which can really get us to this kind of an answer. And so the strategy is to edit the causal variant and assess the molecular change. But there's some challenges. The first challenge is that the efficiency in primary cells in particular of editing is really, really low. So a lot of times you think you're editing in a pool of cells, but you might be editing only 10% of cells. A second challenge that I think people don't talk about that much, but I'll explain this in a little bit, is that a lot of times when you're editing in a pool of cells, you might be seeing the intrinsic effects you want to see, but you might be seeing a lot of extrinsic effects. You might be seeing a lot of effects just because you're perturbing a pool of cells. And so editing SNPs doesn't always lead you clearly to the answer. So our solution has been to develop a multimodal strategy with targeted genome sequencing of the edited locus. So here's the idea. The idea is that you've got a SNP, you want to edit it. If you think the SNP is, say, an EQTL, if you think it's causal for an EQTL, if you think it's going to affect gene regulation, what you'd expect is that when you splice in, put in the T allele, that you have an increase in expression. That's what we want. In practice, what's happening is you have a pool of cells. You're editing maybe one of those cells in among 6 or some such proportion. And so, yes, you're editing not all the cells, but a subset of them. And that's leading to differential expression in the whole pool of cells. So the changes in expression you see are a mix of the intrinsic effects in red, but also extrinsic effects in blue. And the extrinsic effects are sort of like confounding your signal. So to get at this, here's a motivating example just to illustrate the problem. So this is an EQTL for a gene called RPL8. It's not a disease allele, but I think we just wanted to use it to express the point. It's so you have this particular SNP. You can see that as you get more and more T alleles, you have lower expression, more C alleles, higher expression. And if you take a whole bunch of EQTL studies, so these are studies where the SNP has been associated with gene expression, you take all your association studies, you can see the top SNP is always the same one. If you apply a fine mapping algorithm, you end up with this one SNP that you think must be causal. So if you do a simple experiment where you take cell lines, split them into 2, edit half of them, have non-targeted controls in the other, and you look at RPL8, what you see is when you edit the SNP, you don't really see much of a change. So even though you think you have the causal SNP, flipping the base doesn't show the effect that you think you should see. On the other hand, you see a lot of these extrinsic effects. If you do differential expression between your non-targeted control and your edited cells, you see a lot of genes that are impacted because, again, you're seeing these like non-specific effects on the environment. So our solution has been to use this method called, we used to call it Minecraft-seq. Some of the reviewers at Nature told us that it might interfere with the video game that people play, I guess. So we had to change the name of the method. It's called Craft-seq now. And then the idea is that what we do is we take cells, we sort them one by one after editing. We sort them one by one into these 384 well plates. And then we're able to assay the RNA. We're able to assay surface marker proteins. And very importantly, we're able to sequence the DNA. So we know exactly which cells got the edit, which cells didn't get the edit, and which cells got random sort of bystander edits that we don't want. And now what we can do is we can look in the pool of cells what we edited, and we compared the ones that got the edit versus the ones that didn't. So that gets us a very good single cell resolution view of edited versus unedited cells, looking at all the cells with the same genetic background in the same pool of cells in the same environment. So this is that same SNP again. Now what you see is that we go ahead and try to edit, and down at the bottom on the left, you see the controls. On the right, you see the CRISPRed cells. So you see the majority of the cells aren't getting any editing at all. About 194 cells get one SNP edited in, and 26 cells get two alleles edited in. And now what we can do is we can go ahead and look at the gene expression. If you look at the control cells versus the CRISPR cells, this is similar to the pooled experiment we did. You don't see any change in RPL8. But now if you look at the edited cells, the CRISPR cells, and you break it down into those that got 0 edits, 1 edit versus 2, you see that exact increase in RPL8 expression that you'd expect. And now you can actually go and test every gene in the genome, because this is a genome-wide transcriptional profile. And what you see is the SNP's only effect is on RPL8 and on no other genes in the genome. So this actually helps prove that this SNP is actually causal for this locus. So to summarize, we're able to use base editing to define causal variation. But if you're not looking in individual cells and genotyping, you have confounded results because of low editing efficiency and non-specific extrinsic effects. And our solution has been to develop this protocol called CRAFT-seq, which really the key thing is that we're doing DNA sequencing of individual cells to understand which cells are edited and which ones aren't. In the next part of the talk, I'm going to talk about defining state-specific trans effects of the CD40 autoimmune locus. So again, I'm going to go back to this. I'm going to go back to this CD40 locus here, and we're going to focus on this one here. And this project, since the original report of that locus in 2008, you can see that our sample sizes have gotten larger, our genetic studies have gotten bigger and more diverse. But what's really happened is the strength of the association has gotten stronger, but you still see that there's a whole bunch of red SNPs in tight linkage. It hasn't really identified which allele is the causal one. The other thing that's happened is other groups have now looked into other diseases such as multiple sclerosis and other autoimmune diseases, and the same locus has been implicated in multiple diseases. So the evidence of association has gotten stronger, but again, each time you see this string of red SNPs that all have the same evidence because they're, again, all in perfect linkage with each other. So this project is the work of Yoshi Tomofuji, who's a postdoc in my group. And it really started when he started looking at site-seq data. This is single-cell data where you have surface marker protein data. And he started to ask, are variants associated with surface protein expression of individual proteins? And he put a data set together of 1 million cells. And he's clustered the cells into 8 major cell types, so CD4 T cells, CD8 T cells, B cells, monocytes, etc. He found that many alleles were associated with expression of surface marker proteins. So he found 195, we call them SPQTLs, or surface protein QTLs. These are SNPs that are associated with the expression of PD1, of CD4, and so on and so on. About 74 proteins were implicated across 8 cell types. And there were some interesting things that we saw. So a lot of times when we see a SNP that's associated with gene expression, we expect that that SNP is also going to be associated with protein expression. That doesn't always hold together. So this particular SNP, you can see is associated strongly with KLRG1 expression. It's a strong EQTL, but has no evidence of protein expression. So that's one example of a discordant situation. So now back to this CD40 locus. So on the right, you see our RA data, our disease data. On the left, you see the CD40 protein expression and the associations there. And you can see the pattern really does look very much the same. So the same SNPs that are driving CD40 protein expression on B cells are the same SNPs driving autoimmune disease risk. Very interestingly, if you look at expression, gene expression, the SNP is associated with protein expression, but not with gene expression, which is a little bit unexpected. One thing for those of you who are experts on this gene would know is that CD40 is expressed on both B cells and myeloid cells. So at the top, you see CD40 expression mostly in B cells on the right, not so much plasma cells. And if you look in the bottom in myeloid cells, you can see there's a subset of myeloid cells for which you see CD40 expression. So we did have to think about myeloid cells. We couldn't just assume that this was a B cell signal. What's really interesting is if you look in the lower right, you see the pattern of SNPs and its association with CD40 and myeloid cells. And you can see the lead SNP is a blue SNP, not the red SNP. And at the top and the upper left, it's the red SNP. So the SNPs driving CD40 protein expression in myeloid cells is actually different than the SNPs driving CD40 expression in B cells. So that led us to believe that the effect of the SNP in disease has to do with CD40 expression in B cells and not other cell types. Here's kind of what the data looks like. So if you look at the top SNP and you look at your population data, you see really no association with gene expression on the left, about a 20% reduction in protein expression with each allele of CD40 that you have. And again, a very similar pattern between RA disease and CD40 locus. So the next question was, which of the multiple possible SNPs are really driving the signal? And if you look at this locus, if you look at this is the RA data, you're looking at the CD40 protein data looks the same. You can see there's a whole bunch of SNPs with equivalent statistical evidence of association. You can narrow down the scope a little bit if you remove those SNPs that are sitting in an open enhancer or are sitting in the CD40 transcript. You end up with about 4 SNPs, which might be driving the phenotype, but you still don't know which one of those 4 SNPs is ideal. So again, now we go back to CraftSeq and say, can we prove this mechanistically? Obviously, there's a lot of methods out there that can predict one SNP over the other using computational biology approaches, but that's not nearly as good as trying to do this functionally. So we went ahead and applied CraftSeq. Now here we've actually edited each of these different SNPs. And what you can see is the SNPs have no effect on CD40 expression, which is not unexpected because none of these SNPs are associated with CD40 expression. But now let's look at the protein data. On the left, you can see when we edit in a knockout, you see a dramatic reduction in CD40 protein expression. 3 of these SNPs have no effect on CD40 expression. So even though these are all in linkage with each other, they're not actually doing anything when you edit it into the same genetic background. But on the far right, what you can see is that as you edit this particular SNP in the 5' UTR, you see that 20% reduction in CD40 expression, very consistent with what we see in the population data. So to us, this seems like this is probably the SNP driving the signal. This is the same data in another way. Again, here's our edited cells. You can see we're looking at sort of hundreds of cells. We don't think it's a power issue because you can see that we see the effect in that bottom bar where we have actually fewer cells than some of the other conditions. And you can see that the effect is highly statistically significant. What we think the mechanism is this particular SNP sits right around in the 5' UTR, right in the Kozak sequence. So we think that this SNP is actually disrupting translation and not transcription. Our intuition for a lot of the SNPs that drive complex traits is that they're altering gene regulation, altering a promoter, altering an enhancer. But in this case, that's not what's going on. This SNP is actually disrupting translation, and that's what's leading to this reduced CD40 expression. So can we really define trans effects? So trans effects, as many of you who follow this field will know, they've been really hard to find in population data. We're often able to find these SNPs associated with disease. Sometimes we're able to show that the SNP is associated with an individual gene. We're rarely able to show that SNP actually affects something bigger in a cell, multiple genes, or an expression program. But we thought maybe this would be a good way to do it, actually by editing the SNP. And now knowing what the causal SNP is, we can pursue this kind of an approach. And this is because we do the editing on an isogenic background. Cells are all in the same shared environment, and we can actually model the disease-driving cell states. So if you do what many have done, if you take the SNP and you take a big population of blood single-cell data, and you say, is this SNP associated with expression of any genes? What you see is there's really no effect at all. Again, this is your sort of population-based trans-QTL data. Very hard to see any kind of an effect. we decided to take a different approach. Again, trying to take this editing-based approach. This time we took data, primary B cells, from 3 different individuals. We had individuals with the 2 different genotypes. We edited them in both ways, taking CC individuals, pushing them towards TT individuals, pushing them towards CC, just to make sure we were seeing a kind of consistent effect. And then when we applied our Craft-seq protocol to these primary cells that we culture with CD40L, what you see is that we get a spectrum of B cells because some of them are differentiating. You see activated naive B cells in the earliest stage, intermediate and fully developed plasma cells on the far left. And then we can ask, what's the effect of the SNP in each of these different groups of B cells? So if you look at the CD40 protein expression, you can see that the average expression of CD40 of the protein is like different in these different B cell states, but the effect of the SNP is pretty consistent. Like wherever the SNP is, it's always resulting in a reduction of CD40 protein expression in a very significant way. But what about the other genes? And this is where it gets interesting. So what you see is that the SNP, which is having a profound effect on an important signaling molecule, doesn't have any downstream effects in plasma cells or intermediate cells. But in these activated naive B cells, that's where you see a lot of genes are being impacted. So the effect of the SNP on the cis gene, on CD40, is pretty promiscuous. But the effect of the SNP on hundreds of downstream genes is again seen only in this very specific moment in the B cell's life. You can look and see what these genes are. Maybe not a huge surprise. We see genes like TRAF1 over here. And then if you take all the genes, all those hundreds of genes, you can do a pathway analysis. And what you see is that the CD40 signaling genes known to be involved in CD40 signaling, are enriched among these trans genes. Again, why don't we see these trans effects in blood when we look at SNP associations? Well, if you look for this activated naive signature in B cells in the blood, you really don't see anything at all. This population is extremely rare, totally absent in unperturbed blood cells. On the other hand, if you look at inflamed tissue, here you're looking at inflamed synovial tissue from rheumatoid arthritis patients. You can see in the middle, there's a big green blob of B cells. That's a sort of a lymphoid aggregate. And you can see a subset of those B cells have this activated signature. It's not all the B cells, but a subset of them. So what we think is that the effect of the SNP is very specifically happening in this very specific cell state, which is seen in inflamed tissue and is playing an important role in inflamed tissue. Again, giving us the idea that activated naive B cells are a key state in the development of rheumatoid arthritis and the key cell state where this particular SNP acts. So all that was with editing cells. Another thing we can do is we can take individuals, sort out their B cells from the blood and actually perturb them with CD40L, stimulate them in exactly the way we did in the dish. If you take CC individuals and TT individuals, again, you see what you'd sort of expect, which is at baseline, no effect. At 4 hours, 24 hours, no effect. At 72 hours, you suddenly start to see differential expression among these trans genes so that the TT individuals are upregulating the trans genes, the CC individuals are downregulating them. So again, you see them, but you only see them after at a certain key moment and a certain key condition. To conclude, there's an acute need to define B cell states in autoimmune disease. Again, given the exciting results that we're starting to see from CAR T cells, we really need to understand what role B cells are playing in autoimmunity. Defining the causal variant and its function in CD40 may lead to insights about B cells, given the importance of this locus. And the strategy we've been using is this Craft-seq protocol, which we think is very accurate in linking genotype to molecular phenotype data and single cell, single base resolution. To understand CD40, we used this surface marker QTL study, and that helped us define B cells as the driving cell type. And then once we had the causal allele through editing, we understood that it altered protein translation and not gene expression. Then with the power of knowing what the causal allele is, we were able to define context-specific trans effects by editing primary cells. And the downstream trans effects of the C allele occur in activated naive B cells. And the activated naive B cells, we see that they're present in inflammatory tissues, but absent in blood. And this is kind of consistent with efforts to look for trans effects in population studies where we largely don't see them for disease SNPs. And understanding precise B cell states might help us develop therapies that are safe and narrow for autoimmunity and possibly curative for autoimmunity. So finally, I just want to acknowledge a few people. Yoshi has been the driver for a lot of what I've shown you. Yuri and Phoenix developed the CraftSeq protocol, which has been sort of the workhorse here. Kaz and Sauri are former postdocs, now independent PIs, who did a lot of the RA genetics work that I showed you. And Dylan has been helping us understand B cell states and annotate B cell states. And with that, I'll stop and take some questions. Thank you.
Thank you for the wonderful talk. We have 4 minutes for the questions.
Yeah.
Do you have enough matched protein and RNA data so that you could actually do a formal GWAS on the discordance between the protein and the RNA to understand the mechanism or the lack of, or the mechanism of post-transcriptional control?
Yeah, it's a great question. So, I think we're really lucky here because we're looking at surface protein data. So the good news and the bad news is that probably in terms of sample size, there's a lot of site-seq data on blood and, a little bit of, political wrangling can probably get it all in one place. So I don't think it'd be that hard to do like 1,000-person or larger study like this. The downside, of course, is that surface protein QTL data, you're restricted to things for which you have antibodies that stain on the surface for. So that's really, sort of on the order of 100 proteins that you can look at. So I think we could do a pretty comprehensive study in terms of sample size, but not really a pretty comprehensive study in terms of the number of proteins.
But for your CD40 protein of interest, have you done that specific GWAS for the discordance between the RNA and the protein?
So there's zero signal at the RNA level and strong signal at the protein level. So it's an example of a discordant signal. It's really interesting because on monocytes, it's not discordant. So in monocytes, there's a specific expression QTL, which translates to a protein QTL, but it has nothing to do with the disease. So go figure.
Yeah, so really great talk. I'm just wondering for those SNPs that do not change the gene expression level, could it be possible that they are simply disrupting the function of the genes so that they could still be causal like alleles, right?
So, I think for the SNPs that we edited, we really think they have no effect at all. Now,
because they're now in the coding regions.
So they're not in the coding region, they're outside the coding region. And when we edit the SNP that we think is causal, it recapitulates the observed effect. And then the other SNPs, we don't see any, not only do we not see any effect on CD40 itself, we also don't see any effect on any other gene in the genome. So they're looking like very null SNPs. It's possible that if we made our study 10 times bigger, maybe you would see some modifier effect, but for now we don't see anything.
Thank you.
Quick question. So, excuse me. In your study, can you estimate the relative contribution for the SNP in cis and trans effect in total? The single SNP contribute 80% by cis and 20% by trans. Can you reach conclusions, something like that?
It's a great question. It's not something we've tried to do. I think we'd have to think about it a little bit. But certainly we could do it. I mean, sort of maybe the general trend that we see is that the effect of the SNP on cis is most of the effect, except in activated naive B cells, where the SNP is having a pretty big effect on CD40, but it's having a larger aggregate effect on hundreds of genes. So how that shakes out, I don't know. but I wouldn't be surprised if in that specific state that the trans effects are in aggregate larger than the cis effects, but we haven't quantified it.
Great. Let's thank Dr. Soumya again. And I'm very glad to present the certificate of the recognition for his contribution to the field. Let's move to our third speaker of this session. And yeah, so we have Dr. Chen, who is Associate Professor of Stem Cell and Regenerative Biology at Harvard University and a core institute member at the Broad Institute. And his lab is working on developing technologies that bridges single-cell genomics with space and time. Mostly the intersection of the AI genomics, synthetic biology, and the computation to understand where cell types sit within the tissues and also how when their transcriptional programs are active. Please welcome Dr. Chen.
Thank you. Oh, yeah, thank you. I guess, oh, yes, you have the computer.
Do you need to use the presenter mode? Just regular mode.
Regular mode is fine, I think. Yeah, thanks for the introduction and thanks for having me here. I guess I'll just get started while the slides are being set up. You know, my lab develops a lot of, I guess is known for developing a lot of genomics tools to measure tissues and gene expression in the context of tissues. I'll talk a little bit about that today, but I think another challenge that we're often really struggle with in biology is that many of our measurements are destructive. They're like, you know, you have to kill the sample to learn something from it. And so we've always been curious about how do we like look, you know, I mean, obviously you can sample multiple times, but given that also has its own challenges, but given that measurements are destructive, how can we take advantage of destructive measurements and look backwards in time? So a lot of my talk today will talk about that. But I actually just want to start with a different kind of a humorous but also related part, which is I'm a big fan of science fiction, and it'll come up in my talk. But given where we are today, I wanted to start with this article that was published almost exactly 30 years ago, sorry. When they were just sequencing the human genome, it was a perspective by Eric Lander, and they had only sequenced a very small amount of the genome. And I think if you haven't read it, it's very interesting to read in hindsight, because he makes these predictions about what you can do with the genome. And there's not that much on RNA, but there's a couple paragraphs, one of which is saying that, you know, we'd like to one day be able to measure the gene expression of all genes, and that will be informative for understanding cell types and states, the state of the cell, which is actually kind of a mission that we're trying to do now. We can now comprehensively collect single cell gene expression profiles, and now we're starting to understand the programs of the cell. So in some ways, kind of prescient. And then there's one little bit about spatial localization of gene expression. And it says that basically this is going to be important. Obviously, we understand the genes are spatially localized. But it says that DNA arrays would not be suitable for such investigations, but high throughput in situ hybridizations could be feasible. Turns out that, you know, we've spent, you know, our lab has spent the last many years converting basically effectively microarrays into tools to measure gene expression at scale from tissues. And then other labs, and ours as well, has converted high throughput in situ hybridizations to do RNA measurements. So it's simultaneously maybe a little bit off the mark, but a little bit prescient. So where does this relate to science fiction? I think actually in terms of research and science fiction, these three laws are actually kind of related in some ways. And Arthur C. Clarke is a science fiction author, and he famously had these three laws of science fiction, of which this last one most people know, and sufficiently advanced technology is indistinguishable from magic. And this is something that I like to tell people in my lab about aspirations for technology development. And also number two, but maybe this first law applies here. Okay, all right. So that's just a humorous aside. And that's just to say that, so we know for a long time that, this context in which cells, not only are individual cells important, which was really beautifully highlighted by the previous talk, but the context of those cells within tissues is also important. And for a long time, our molecular measurements and maybe many molecular measurements today are still focused on dissociated cells. And so, and so my lab has spent a long time building genomic tools that bring transcriptomes or DNA sequencing directly into the context of tissue, so you can measure the expression of genes and know where those cells live, and then maybe the communications and the relationships between those genes. And also for genomes, that allows us to understand the clonal relationships between cells, as well as the evolution of which I'll get to in a second. But I won't really talk about technologies today, reckoning this third law. I'll just mention that we want to use these. I'll show you how we're using these technologies, except for the fact that I have to introduce what the technology we're using is really quickly, which is that for a long time we developed most of these papers were powered by these high resolution bead arrays where we put microbeads onto the surface of glass slides, and these microbeads basically allow you to capture DNA or RNA, and they act as pixels for in a camera where you're, instead of capturing pictures of photons, you're capturing pictures of molecules. And this has been commercialized, and lots of people have been using this technology. It's called SlideSeq, but there's also a very similar technology called Visium and Visium HD, and this is a very popular way to collect this data. But in my lab recently, we've developed an adaptation of this work that we found to be much more powerful in many ways, in part because these pixel-based approaches are really subject to mixing between cells. And so you have the spatial context, but you no longer lose this, you lose the individual cellness of the data, which turns out to be quite important. So we developed this approach a couple of years ago where we can take any single cell approach and then sequence the single cells and know where their cells came from. And that's quite important because you can start making measurements that are not just mRNA, measurements of the genome, the epigenome, as well as it's much, much more sensitive. And so how it works is that we can take tissues and we put them on the same arrays, but we kind of invert the problem. Instead of trying to capture the RNA onto the tissues, what we do now is these arrays have barcodes, DNA, that basically are photocleavable, and each one of these beads marks the location of a barcode that's being, a population of barcodes that's being released. And when they're released, they go into the nuclei of cells, and they get attached there non-specifically. And then when you sequence it, it's akin to GPS for the cells. Each cell picks up the distribution of local nearby barcodes, and you can then actually use that to fit the location of where those cells came from. And so then you can take any single cell measurement technology. Really, we've tried it for many, many things now, and you can just collect your single cell data, and then at the end of the day, you know where cells came from. A lot of the data in the paper that was published was done with 10x. And so, and this is one of the first data sets in the mouse hippocampus where we did it. And I won't, talk about this today, but you can read the paper, is that, the key is that it's truly single cell data, and it's about 10-fold more sensitive than these array-based capture approaches. And we have a lot of benchmarking data that shows that. And I think importantly, it adapts to genomic and epigenomic measurements. So I just wanted to introduce this because it'll become later for some of these measurements we're making for the history of cells in the primary part of my talk. So here we'll get to the meat of it, which is that, as I've already mentioned, molecular measurements are destructive snapshots. Like, to sequence a cell, generally you have to grind it up and sequence it. And same for tissues. It's you can only sample the tissue from a person once. You can't, for example, this is actually an H&E of a Hodgkin lymphoma sample, right? But in the past, that was probably a ball from a healthy lymph node. And the evolution of the tissue, both in terms of its lineage, but also in terms of the cell state transitions and the exposures, probably tell you a lot about the pathogenesis of disease. But the other thing is that much of what we know about causality, at least in the past, comes from temporal causality, right? How we learned about physics is that we observed thing A happening and then we observed thing B happening, right? And they're related in time. And if you only have snapshot measurements, it's very hard to go and get temporal causality. So it's another reason why we'd be interested in this. And so, so thinking about it, when we're making measurements, you can imagine that the history of cells can be read out through many different molecular modalities. Inherently inside of DNA, there could be mutations or SNVs that encode the clonal structure of cells. Inside the epigenome, maybe there's some history of the exposure and gene regulation. And then transcriptomes are kind of a very short-term memory of the cell state. But I'll tell you today about how we're building that to be a long-term recording of cell state. So we've really been making measurements for these complementary windows into the past. So maybe we can start with DNA and lineage. And that many folks have put a lot of work into trying to understand if we can use DNA for lineage tracing in single cell or in other contexts. And the key is that basically, every time your cell divides, for a normal cell, it accumulates some number of single nucleotide variants. That number is very small out of 3 billion bases. It's anywhere between like 3 and 100, right? So it's like a 1 in 100 million or 1 in a billion measurement. So it's hard. In cancer, there are lots of other chromosomal aberrations or duplications, translocations that you can use as like clonal measurements of lineage. And so many years back, we modified this like slide-seq or spatial array-based capture approach to measure DNA to infer clonal architectures of human tissues in cancer which have large CNVs. So, and that's very doable with any like, you can infer, CNVs are a way to trace somatic evolution. But the challenge is that most healthy tissues don't generally have CNVs, and many cancers themselves don't. And so more recently, we've been trying to do 2 things. One is, which I'll talk about now, is trying to use engineered evolving barcodes in model systems where you can put those engineered evolving barcodes in. And the second is to just use the genome. And that's a hard challenge, but I'll show you that we're getting there. And so actually in 2 separate collaborative projects, we've been trying to take these Cas9-based barcodes, which take a given locus and progressively edits it. So you can use that as an evolving barcode. And we've been trying to read it out in the context of cells. And so I'll just highlight this project that we've been doing with Jonathan Weissman. Here we have a mouse model with one of these evolving barcode tracers, and then you can induce the formation of individual tumor clones in the lung from single cells. And then you can use these spatial genomics approaches to read out both the mRNA as well as this tracer RNA, which encodes the history of lineages in the cell. And then, you can both measure this in the primary tumor because it's seeded from a single cell, as well as the metastasis. And so that you can understand how does lineage map to spatial context during tumor evolution. And so what does this data actually look like? So here, this is a cross-section of a lung taken on one of these spatial transcriptomics arrays. And each colored grouping is basically like one tumor that's seeded by a single cell. And if you just zoom in, if you read out this tracer mRNA, you can visualize basically the lineage relationships of all the tumor clones inside this red grouping. But you also have the spatial context, and you can walk down this clonal tree to look at the subclonal evolution, right? Like basically you have the seeder cell, and then that it is the clone, but then there's local subclones. And it turns out that if you just look at the spatial context of these local subclones, they have very much like strong spatial coherence because they're growing and dividing locally, except for, you can see this, interestingly, subclone 1 has this wide dispersion across the tissue. And so there's many things you can do with this, but maybe I'll just highlight a particularly interesting experiment that we did, which was we took basically these lungs and we sampled them in many serial sections in Z, so we could capture all of the primary seeding clones. And then we also took the metastases and captured them. So we know what the lineages or the lineage barcodes of the Mets are. And then if you look, and so this is one section of the primary, you can look and see basically the allelic distance to the metastases. So which of those cells are actually seeding the Mets. And so in light green here, or sorry, dark green, these cells from this one particular seeded tumor are actually the cells that have the lowest allelic distance. These are the cells that seeded the Mets and many other parts of the animal, actually rib cage, lymph node, and diaphragm. But in the same tumor, right, there are many other subclones which did not seed the Mets. So it's not like this particular tumor seeded the Mets out of a nearby tumor. It's that within a tumor, there's like a subclone that seeds the Mets. And if you look at within, and interesting, so what we did was we looked at what is the gene expression of the seeded cells, as well as the local environment of the seeding cells. And you find that there's a shared microenvironment between the seeding clone and the Mets. They have like a shared hypoxic niche with the same sort of immunosuppressive macrophages, versus even nearby subclones from the same tumor. They have a different microenvironment. So it's like there is this carryover, this, of the microenvironment between the primary and the Met. So in some sense, there might be, they have this shared tissue context. It's not clear if that's they recruit that tissue context, or it's that that tissue context is necessary for that particular subclone. The causality is still unknown, but it's very interesting that they have this shared tissue context, and then they become more collagen rich and fibrotic. Okay, so this is using these evolving barcodes. What about human tissues? You can't like put a Cas9 evolving barcode into human tissues, and especially healthy human tissues. So we've been working, this is a really collaborative project, really led by Andrew Russell, a postdoc in the lab, and Tim Correns was also at the Broad, but he's now at EBI. And so we've been taking this sort of slide tags approach and doing something called Resolvome, which allows you to do whole genome sequencing of individual cells. You sort the cells into plates, as well as single cell RNA-seq. And so I'll just show you a case in cancer and then a case in healthy tissue, how this works. So here is a CRC specimen. And what we do is we take this region and we sorted out about 1,700 nuclei. We didn't sequence all of them. So this is the primary limitation, sequencing costs at the moment. We took about 700, 2 plates of them, and did low pass whole genome sequencing. And when we do that, you can use the spatial barcodes to localize each one of these, about 80% of these cells into the spatial context. So this is where these 800 cells live inside the tissue. And then for a subset of them, we then performed 15x whole genome sequencing, 13 on them. And when you do that, you can use the whole genome sequencing and just the SNVs to cluster the SNVs to derive lineage trees. So here are basically these 182 cells clustered by in the columns, and then the rows are individual single nucleotide variants. And you can see basically there are clear groupings of cells by single nucleotide variants representing locally clonally related cells. And then you can construct this lineage tree, and then where are they localized in space, you can actually walk down basically this tree and see that these major subclones occupy distinct spatial regions. And then as you walk down to finer nodes, you can actually see that the local spatial dispersion is more concentrated, right? Like look at this particular clone here. And so you can start reconstructing the like really fine lineage relationships using these CNVs. And then, in addition to reconstructing the lineage, the DNA also encodes something about the mutational spectra. So this is like what mutations have accumulated, and different processes like maybe like aging or chemotherapy give you different mutational spectra. And so what's actually happening in this sample, very interestingly, is that if you look at the leaves of all of these trees, they have this yellow mutational signature where the cells rapidly accumulate many of these, this yellow signature, which turns out to be chemotherapy. So later on, this patient experienced, so early on, there's these signatures that are basically like apobec or like aging related, where the tumor is evolving. The person got chemotherapy, and you can see this rapid, basically chemotherapy signature that's actually diversifying the leaves of the tree. That actually also helps you lineage trace. It's like an in vivo, barcoding that is coming from the chemotherapy itself. So that's very interesting. So that's CRC. So we also did this in the normal human colon, which is a much, much harder challenge because there's not additional mutations. And so here we took, this is the crypts of the colon, and here's about 370 nuclei that we did 15x whole genome sequencing. We filtered out for, so this is if you just look at the shared mutations, then you can filter out things that are non-epithelial. So there are other cells that are from different lineages, but they're not epithelial, so they don't have shared mutations. But then in these green leaves, you can see kind of these epithelial lineages. And actually here you can see what this actually is that at these upper, these blue segments represent fissions between crypts, and on the bottom, they represent actually like, local divisions within the crypts, and then here are the early embryonic SNVs. And in, this is like, amazingly, right? You can see that if you just mark these like individual crypts, you reconstruct the like local organization of cells inside the crypts inside this colon. So it's like this is like actually lineage tracing in space in a healthy human tissue just from the somatic SNVs. You just have to perform whole genome sequencing on each cell. So we're working on that. Why is this exciting? Well, there's probably, actually one additional thing. So there, if you look at the mutational signatures, there's this particular phenomenon where early on in all of these epithelial cells, so again, you rank the SNVs over the trees, you can know which events occurred early, which events occurred late. Earlier on in all these epithelial cells, they have the signature for colobactin, which is this bacteria that secretes this toxin which causes DNA damage in these epithelial cells. And that's actually linked to colon cancer. And it's been speculated that this particular phenomenon happens in early childhood. It's like some weird, like local transient microbiome, but you can actually see that in the data. And that in this person, they only had this colobactin signature early on in these lineage crypts. And so why is it exciting? Well, I think there are lots of tissues in humans where we'd like to be able to understand how they evolved in lineage as well as their spatial architecture. And so, this is probably one of the first times we can start doing that in healthy human tissues. So we'd like to be able to expand that to many other contexts. Okay, how much time do I have actually? Do you guys know? 5 minutes? Okay. So the other thing I said, so that's DNA. The other thing I said is we'd like to be able to understand the past transcriptomes of cells. And this is a dream of ours for a long time, which is that, you often see these maps of fate transitions in cellular differentiation, where you have this idea that in the past, cells were the same, right? They chose two different fates, but in the past, they arisen from some shared state. And what was different about this past cell state that biased them towards future states? We might think that's gene expression, we might think that's epigenetic or it's stochastic. But what is it? And so, we've always wanted to make a time machine for cells. Given where you are in the present, because I can only see you in the present, what was your state in the past? So if, was there any difference between the gene expression of cells that went on to become fate 1 versus fate 2? And so this seemed like a really hard problem. Actually, we've been working on it for a long time, and mostly it seemed impossible. But we actually found this particular thing in, we had this inspiration. We tried to do this for a long time. We had this idea that in elementary school, some of us would put stuff into this thing, time capsule, and you would bury it in the ground. And then later on, you can come out and retrieve it, and you get to know what you were doing in elementary school. So can you get the cells to do that? Bury their transcriptome, and then later on, you can retrieve it. And we tried a lot of different things, and then eventually we settled on this thing called the vault particle, which is this enigmatic organelle that exists in human cells already. It is this huge complex of 78 major vault proteins 39 on each half, and it's basically hollow. It's like a shell. And it can fit about 2 ribosomes inside. And it turns out that what we realized is that you can turn this into a time capsule by fusing a poly-A binding protein that attaches onto the inside, and basically mRNA just get captured and sequestered inside. And then it becomes an RNA storage device. Sounds kind of crazy. So it turns out that when we do this, basically when we express basically this major bulk protein along with the poly-A binding protein, you can capture 1 to 3% of transcriptomes inside of human cells, living human cells. And it's very specific to having both the bulk and the poly-A binding protein. And the capture is very representative of the transcriptome. It has really high fidelity, correlation of 0.95. And it's really non-perturbative of the cells. You can get thousands and thousands, about 20,000 volt particles per cell. In fact, some of your human cells already have, your epithelial cells, some of them have, by staining, tens of thousands of volts already. And so, it's very non-perturbative. And then the key thing is that when you put the things in the vaults, it stabilizes them. The mRNAs can be protected from RNases in vitro for about 2 weeks and about a week in vivo. So it is basically an inert capsule for the mRNA. And then what does that mean? It means that you can do the experiment I told you about, which is we put this thing under Dox induction. You store the RNA in the cells, right, this time capsule, and then you wait, and then you can retrieve the state later. And so I'll just show you an example of what that means in cancer drug resistance. So basically, you can treat cancers with targeted therapies, and then the cells die, but some subset of them sometimes survive. And we think that maybe there's some pre-existing state that is drug tolerant. And so what is the molecular state of those cells? And so, for example, here in PC9 cells, when you treat them with an inhibitor for EGFR, about 10 or 20% of the cells always live. And it's a non-genetic resistant population. And so then the idea is that we would perform recording in the past, and we would sample the cells that lived versus the cells that didn't live, the cells that never saw any drug, and ask what was different about their past transcriptome. And so when you do that, you record the cells, you can see that the cells that lived have upregulated OXPHOS and downregulated, they're basically more quiescent. So that's kind of well known, but it's still a very validating result. But if you look at then genes that are really, so this is gene ontology. If you look at genes that are actually the most upregulated, you see many candidate genes that are in ECM adhesion, autocrine signaling, metabolic enzymes that are pre-existing in these persisters, specifically when you record them, that bias them towards living. And actually, one of these genes, fibronectin, you can stain for it, and you can see that there is before you treat with the drug, there's a small population of cells that are fibronectin high. And then when you treat with the drug, you select for this fibronectin high population that is in the persisters. And so we actually performed pharmacological and transcriptional perturbations of all of the top of 5 different pathways. Here I'm showing you this very interesting result with siRNA knockdown of fibronectin. If you just treat with the drug, about 20% of the cells live. If you knock down fibronectin, it has no effect on cell viability. But if you treat with both EGFR inhibitor and knock down fibronectin, you see that you eliminate the persister cells. You actually prevent these cells from surviving the drug. And so the idea is that we're performing this temporal differential expression. And so the causality, so you can make hypotheses about what things are maybe causal for these cells to be persisters. And so I think I'll just end here. I think these cellular histories can be read through many different views. We've been looking at the genome. I didn't talk about the epigenome, but we're quite interested in that. And then also these molecular memory recorders. And the reason is that maybe past states are natural perturbations that help separate cause from consequence. And I think I'll just end here then. Thank you for your attention, and let's understand.
Thank you for the very great talk. So now we may have one question.
Oh.
Actually, can I ask two questions? So first is a random one. So Faye, do you have a, did you write a time capsule when you were a kid? And did you write, you want to be a scientist and you want to make sci-fi into reality?
I don't think I did a time capsule when I was a kid, although, yeah, I think I know, maybe I can think about what I would have written. I don't think I would have written I would have wanted to be a scientist, probably an astronaut or something.
Great. Okay, so second question. So what are the limitations we know about the vault? So for I think the limitations we want to know, for example, do they reside in the cell at different locations that may or may not bias their capturing of the transcriptome? And what are the total amount of transcriptome that will be captured along the 1 week or 2 week time?
Yeah. Those are great questions. So I think they're primarily cytoplasmically biased. Actually, if you look at the genes that are differential between the lysate and the vault captured, it's stuff like nuclear genes and mitochondrial genes. They can't capture things that are not in the cellular compartment that they're made in. And then, we can only, right now we're capturing a couple percentage of the transcriptome over weeks. And so how we're thinking about this problem is it's like a pseudo-bulk problem because, you can get the cells to choose a fate, right? And you can take all the cells that chose that fate. And here in the persister cell case, it's like whether they lived or died in the drug. And then you can look backwards in time. And so the capture efficiency just limits you towards how many cells you need to do the experiment.